All right, so once we have formed our various T cells, right? So in the last part we talked about making T cells, CD4, CD8. Um, they will ultimately become activated, and we'll talk about how they become activated here in just a few minutes. And when they become activated, they'll undergo this process of clonal selection, right? So clonal selection is simply the idea of a cell after it responds to an antigen and it becomes activated, it's going to divide. So it's going to undergo mitosis and it's going to produce more copies of itself. Right now, the most important thing related to this clonal selection is the fact that the original cell, when it divides, all of those downstream cells, all those new daughter cells, they have the same specificity as the original cell that was activated, right? So they retain their receptor specificity. And so this is this idea that we talked about in the very first part of this adaptive immune system lecture of immunological memory, right? So they're keeping a memory. They keep the same receptor specificity for the same antigen as the original cell that was exposed to an antigen and then divided. Right. So what stimulates this clonal selection is an antigen challenge. So this is the encounter between an antigen and a lymphocyte, usually in one of those secondary structures, right? So lymph nodes, spleen, tonsils, mucosa-associated lymphatic tissue, something like that, right? So what we can look at is we can look at how you actually activate your T cells. Okay, how you actually activate your T cells, right? So you can read through this description here. So how do you activate it? First signal, second signal. I'm going to show you a image because I think it helps it make a little bit more sense, right? So our first signal is when the CD4 molecule binds with the MHC molecule. So when you see these terms, automatically ask yourself which cell type has each of these, right? So where do you find the CD4 molecule? You find that on the T cell, right? What type of T cell? That's a helper, right? MHC2 molecule, where do you find those? You find those on your antigen presenting cells. So macrophages, dendritic cells, B cells, things like that, right? So the T cell receptor is going to interact with the MHC molecule on the antigen presenting cell. So you can see that interaction right here. It's going to be stabilized in part because of the CD4 molecule. And then, so that's the first signal, and then that will then initiate the T cell to begin secreting and producing these cytokines called interleukin, right? Interleukin 2 in particular. So interleukin 2 is what you can think of as an autocrine, right? An autocrine. So remember, there's different types of signaling molecules. There's endocrine, there's paracrine, there's autocrine. In this case, an autocrine signaling molecule is a molecule that stimulates the same cell that released it, right? So this T helper cell, this helper T cell is activated. It secretes interleukin 2, and interleukin 2 binds back to receptors on the helper T cell, and then that helper T cell begins to divide, right? It begins to proliferate in that process of clonal selection, right? So meaning it's making clones of itself, it's making identical copies of itself, right? Now this was a naive helper T cell, so meaning it had not yet encountered an antigen. And then whenever it divides, although the receptors are the same, you call the cells something different. So you may call them an activated helper T cell and a memory helper T cell. Right? So the activated helper T cells, those are the ones that are actually initiating the immune response, right? So they're going to help with both the cell-mediated immunity and antibody-mediated immunity. That we'll get to in just a moment. The memory helper T cells, these are going to kind of stick around. These are going to remain in circulation for periods of time. And in the event that they encounter the same antigen, those memory helper T cells are now stimulated and they further divide into activated helper T cells and even more memory helper T cells. Okay. So that's what's happening with helper T cells, right? How they become activated. How do you activate cytotoxic T cells? It's a very similar process, right? So you have a first signal and you have a second signal, right? So first signal again, you have your CD molecule interact with the MHC molecule. So remember, CD, that's on the T cell, cytotoxic T cell. In the MHC type 1, that's on the nucleated cell, right, including antigen presenting cells. So it could be antigen presenting cells. It could also be an infected normal body cell, right? So the T cell receptor on our cytotoxic T cell is going to bind to and recognize the foreign antigen being presented to it on the MHC class 1 complex, right, on that MHC1 molecule. That's the first signal. And then the second signal, there are other receptors. So there are other CD molecules. There's not just CD4 and CD8. There's CD28 and there's others. And those others are going to ultimately help to stimulate this cytotoxic T cell. And then when this cytotoxic T cell becomes activated, it's going to divide into activated cells and memory cells. So just like the helper T cell, activated helper T cell, memory helper T cell, you get activated cytotoxic T cells and memory cytotoxic T cells, right? So one of the stimuli for these cytotoxic T cells to become activated are the interleukins from the helper T cell in the last part, right? So if you look back here, here's our helper T cells becoming activated. You can see here they are beginning to produce these interleukins. And so this arrow that you kind of see coming down here that's cut off, this is the continuation of that arrow, right? So these interleukin 2 molecules from the helper T cell act to stimulate the cytotoxic T cell for it to begin that process of clonal selection, right? So now it's dividing into activated cytotoxic T cells and memory cytotoxic T cells, right? And the cytotoxic T cells, we'll see in a little bit the effect. They're the ones that actually end up destroying the infected cell, right? And we'll talk about how that works in just a moment, right? And then you have your B cells, right? So we can't forget about our friend the B cell, right? So in this case, you have similar things, first signal. Now in this case, the outside of the cell, in this case the B cell, its receptors are the antibodies, right? The antibodies are going to bind the antigen, okay? And remember, the B cell itself is an antigen-presenting cell, right? So it will bind it on the outside. It can engulf it, bring it in, endocytose it, process it, present it on its own surface MHC molecules, right? And specifically MHC2, because this is an antigen-presenting cell. And then an activated helper T cell, maybe from the previous step, right? Whenever it became activated, those activated helper T cells can now bind to and recognize these antigens, further stimulating the B cell, and then the B cell is going to divide into what's called a plasma cell, right? A plasma cell. And again, these are mediated through those cytokines, the interleukins, right? In this case, it's interleukin 4, all right? So here are your B cells. When they divide into a memory B cell. Again, the receptor is the exact same as the original, right? It retains that receptor specificity, right? So it activates the B cell to divide into memory B cells, and then these plasma cells, right? So this is the same image down here. So these plasma cells, these are what are actually producing the antibodies. So think of the plasma cells as like antibody factories, right? Antibody producing machines that are secreting out a large quantity of antibodies. And again, these antibodies have the same specificity as these on the memory B cell, as the ones on this inactivated B cell, as the ones on this original naive B cell, right? The receptor specificity stays the same throughout the entire process, right? So these antibodies that bind this particular antigen, that are bound receptors, these bound antibodies, these freely circulating antibodies, they would bind the exact same antigen, right? They would bind the same exact one, right? And so again, the B cell receptors, they're going to stay the same. You have the memory B cells, and again, when these memory B cells become activated in the future, when they're exposed to the same antigen, they're going to further differentiate and divide into more memory B cells and more plasma cells, right? So what we'll see in a little bit is we'll talk about different types of exposure, so kind of the initial exposure to an antigen and secondary exposures. And as we'll see, the secondary exposure, the second time you encounter the same antigen, the immune response is typically it's heightened, right? It's more drastic, and it's faster as well, because now you already have all of these circulating antibodies, right? And so we'll talk about that here in just a little bit, right? So let's stop here, and we'll pick up in our next part looking at the effector response. So what do the cells actually do? And we've already kind of alluded to that for for some of them.